how powerful is any strategy going to be based on one small molecule? And how well do we actually understand how this network will respond to me playing with the concentration and activity of any one of these parts? And so I asked that as a question, we'll get back to that in a moment. So again, what I'm trying to do is take stock a little bit. So where are we now in 2010? Entire human genome, we've got that. Do we know most of the key proteins? Probably. We easily know 60, 70, 80% of the key proteins in cancer. We at least know their name. We know their structure and function individually. This is true of many things. This is not true of transcription factors. This is not true of many protein remodeling factors. But we have pictures of many of the important participants. We have some general sense of these interaction networks, and they're becoming daily ever more complicated. We're adding, every day, we probably add 10, 15 arrows to these signaling interaction networks. And again, the question is, do we now understand the system well enough so that we can really treat all diseases? Do we understand the system well enough that we can deal with something like cancer, which of course changes over time. During chemotherapy, cancers become resistant to certain agents and so forth, so these systems are changing. And so one of the most famous physicists that you have all heard about is Richard Feynman. And this is a picture of his blackboard the day he died. And so he has any number of, he's enumerated major problems. So to learn, we have a variety of the 2D Hall effect, various other things. We have some more cryptic statements. But one of his most famous statements, of course, is what I cannot create, I do not understand. And what this statement has turned out to be is a very powerful way of thinking, of measuring how much we understand. So if I asked you to create a cell, a biological cell that has all these functions, it divides and it senses chemical concentrations and it migrates through the body, could we, with what we understand now about biology, do that? And the answer is a clear no. And this is true of basically every single aspect of cell biology, of tissue biology, of developmental biology of most diseases. And so it's easy to fool oneself into thinking that we actually know a lot about biology. But now, what we do know in 2010 is that even after we sequence the human genome, some of the very, very basic mysteries are almost as open as they were 50 years ago or 100 years ago. And this is all based on the standard, what I cannot create, I cannot understand. And so why do we have these deficiencies? Why do we not already understand biology, disease, regenerative medicine? And is this a simple matter of scale, cost, infrastructure, dedication, lack of interest? So if all we did is we threw a lot more money at this problem and waited a little bit longer, would we necessarily, if we drew another 1,000 arrows into a signaling network diagram, would it then become clear? What if we threw in another 1,000 proteins and another 10,000 arrows? And so what I think personally this has to do with is a rather significant lack of basic biological organizing principles and mechanisms. And so if you think about the Lego box, I have a little kid, and so his Lego box guaranteed never looks like that because every single Lego is spread out over the floor. But we are very good as in biology of characterizing all these individual parts. But what we really want to do is we want to see how all those individual parts work together to bring about some biological function, like appropriate cell division or inappropriate cell division and so forth. And what I'll do now, I'll just briefly touch on sort of three very, very general problems in biology that we'd like to understand, but we don't. And you're already in the fortunate situation that you've already been briefed by one of the best practitioners in this entire mechanobiology area, Valerie. And so a lot of what I'll say soon will be clear. So the first problem that strikes me that we don't understand very well is relating genes to particular cell types. And let's do a quick order of magnitude calculation. So the human genome has 23,000 genes, right? These genes are not like Lego blocks because what we know is that each of these genes, each of the proteins produced by the genes is subject to exquisite regulation. And so each of these objects, these proteins, its location in the cell is controlled, it may be chemically modified, and it is also, just one aspect of this regulation, let me touch on, is splicing. And so here's the idea. For every single gene, we typically have several different coding regions denoted by these colored arrows called exons. And so for every single gene, whether it's P53 or any protein that you know about, I can make many different proteins. So I can take the yellow part and the red part, or the yellow part and the green part, or the yellow part and the blue part, or the red part and the green part and the blue part, or not the green part, and so forth. So every single of those 26 or 23,000 genes can, in principle, give rise to many, many, many different proteins, each differing by their composition. And so now just imagine you have 23,000 genes, but each of them you can make many, many different flavors of, 3, 4, 5, 6. So if you think about this, the number of theoretically possible distinct sets of proteins is absolutely astronomical. So if I give you this blueprint called the genome, then in principle, you can make an astronomically large number of distinct combinations of these proteins. It's a mind-boggling number. It's larger than the number of atoms in the universe. And yet, how many different cell types are in your body? Do you have 100 trillion different cell types? No. What you typically have is on order of several hundred different cell types. Things like neurons and fibroblasts and all the other cell types that make up your body. And so, despite there being an astronomically large number of possible combinations of all those parts, you end up, if you look at yourself, with something on order of 200, 300, 400 distinct cell types. So in the language of physics, this is telling you that the system is highly degenerate. It must be that an exquisitely large number of microscopic combinations all give rise to one thing, like a fibroblast, like a neuron, or something like that. And this is really, and so now this is a first issue that the scientific community really does not understand very well at all. How is it that we have this blueprint that in principle can make an astronomically large number of distinct cells, but if we look at our bodies, we have a countable number. And so one of the other questions is, once we're a certain cell type, let's imagine a fibroblast, or a green fibroblast, how am I able to switch this green fibroblast to some other cell type? And so you can imagine where this is going. Because a lot of problems in disease and regenerative medicine and developmental biology, of course, involve a cell of one type switching to another type, whether it's a stem cell differentiating or something like that. And here's something here's one example of how to accomplish this sort of switching. So this is work that was recently published from a group at Stanford. And so what they did is they started with a fibroblast, which is the cell on the left, and they added 3 things, and they were able to convert this fibroblast into a functional neuron. And this is equivalent to this medieval idea of turning like a lump of coal into gold. And how is it that you're able to take a cell and you add 3 things, and you have a functional neuron with electrical activity and all these things? And so again, this is intimately related to problems in developmental biology and disease. Because what's happening in cancer, for example, is that just imagine you inappropriately were to switch a cell type from one type to another, and so forth. Likewise, if you could understand this code of how to switch a cell from one state to another and back, then you would essentially have any number of new ways of treating disease and so forth. And so this strikes me as one sort of large set of questions that the field would like to understand better. So there's a direct link to cancer of these considerations, and this is something that Mina Bissell and also Valerie have thought about for a long time. Here's a classic experiment. We're used to thinking of cancer cells as being determined by their genome. So once you have certain mutations, you're a cancer cell. But what Lena has done in this case is she's taken the